Good afternoon to all. Today I will talk about a project aiming to study the effect of muscle activity on the association of two intracellular organelles which are crucial for skeletal muscle function, that are mitochondria and calcium release units. Calcium release units are specialized intracellular junctions devoted to excitation-contraction coupling, a mechanism that allows transduction of the electrical signal coming from the neuromuscular junction into release of calcium from the sarcoplasmic reticulum. Calcium released during excitation-contraction coupling activates muscle contraction, and fibers will then relax when calcium is pumped back into the SR by the Sarca pumps. It has been known for a long time that a small amount of the calcium released during EC coupling also enters mitochondria, the intracellular organelles devoted to produce ATP during cellular respiration. The entry of calcium into the mitochondrial matrix via MCU, the mitochondrial calcium uniporter, is metabolically important to stimulate the respiratory chain and to increase cellular ATP production during muscular contraction. However, it has been proposed that mitochondria, to take up efficiently calcium, must be placed in close proximity to sites of calcium release. And our lab has become interested in this scientific issue since when in 2009, Simona Boncompagni and colleagues published a paper describing the spatial disposition of these intracellular organelles. In fast-twitch skeletal muscle fibers, mitochondria are placed at the I-band of sarcomere, next to the Z-line and in close proximity to calcium release units, that in skeletal muscle fibers are also called triads because they are formed by one transverse tubule coupled to two terminal cisternae of the SR. And as Feliciano told this morning, we believe that this spatial disposition is very important for SR mitochondrial calcium signaling and likely for proper metabolic efficiency of muscle fibers, as we found different conditions in which this precise mitochondrial triad association is lost, like denervation, aging, and several myopathies. And today I will talk about aging. In this paper published in 2015, we have shown that in skeletal muscle fibers from aged mice, mitochondria or part of mitochondria lose their correct position. In fact, in this double immunolabeling, we mark the ryanodine receptors in red, while the green staining marks position of mitochondria. And as you can see here, the perfect double cross striation that is visible in adult fibers is partially compromised in fibers from aged mice, with the areas in which mitochondria forms longitudinal clusters. And these electron microscopy images show triads with improper or oblique or longitudinal orientation, and also some mitochondria extending longitudinally between myofibrils. These data were published in this 2015 paper in which to address whether the aging caused the changes also to the association of mitochondrial calcium release units, we quantified the tethers, these small electron-dense strands that link the outer mitochondrial membrane to a terminal cisterna of the triad. And the results of these experiments indicated that the number of tethers was significantly reduced in fibers from aged mice, while average minimum distance between adjacent mitochondrial and SR membranes was significantly increased in fibers from aged mice compared to that of adult mice. In the same paper, we also hypothesized that the age-related elevation on oxidative stress could damage membranes and proteins, and this could in turn activate a feed-forward mechanism and can lead to also activation of proteolysis. So, in the last 5 years, in our laboratory, we have been working on the following scientific hypotheses. The correct association between calcium release units and mitochondria, which is challenged by aging and inactivity, can be improved or maintained by muscle activity and exercise. So we worked on 3 different groups of samples. In collaboration with Helmut Kern in Vienna, Sandra Zampieri, and Ugo Carraro in Padua, we analyzed human biopsies from young adults, sedentary seniors, and age-matched sportsmen who had exercised regularly, at least in the second part of their life, and there were mostly bikers, skiers, and training in the gym. Then we studied also EDLs from mice, either control, aged, and age-trained. And finally, we also evaluated the effect of denervation in adult rats. We studied, in collaboration with a research group in Urbino, EDL muscle from rats denervated for 15 days and denervated plus reinnervated for 15 plus 30 days, a period of time which allow muscle to be reinnervated by regeneration of the crushed nerve. So, using electron microscopy, we quantified in human biopsies frequency, expressed as number per area of section, morphology and orientation of calcium release units and mitochondria. But today, due to time constraint, I will only show you data for the mitochondrial analysis, which gave more striking results. With age, mitochondria volume, number, and also association with calcium release units decreased significantly, whereas in sportsmen, these age-related changes were almost completely prevented. In fact, mitochondrial volume, number, and also association with triads were very similar to that observed in young controls. These data were published in 2015, and this electron microscopy image summarizes changes induced by aging, where both mitochondria and calcium release units lose proper morphology and orientation. On the other hand, in panel C and D, there are images from a sportsman of the same age. But in this case, mitochondria that are indicated by the white arrows are correctly positioned on both sides of a Z line, indicated by this black arrow. And calcium release units have the classical three-element structure, hence the name triad, and are often correctly positioned next to the mitochondria, as you can also see in the magnification panel D. So, to confirm our studies on human biopsies, we started a rigorous and controlled study in animal models to reinforce evidence that muscle activity improves or maintains the correct association between calcium release units and mitochondria. We compared aged and age-trained mice, which had access to training wheel for 1 year, starting at 1 up to 2 years of age. And first, we wanted to verify that our protocol of exercise was able to improve the overall function of aged mice. And we measured in vivo grip strength and ex vivo contractile force. Our results confirm that our protocol of exercise improved muscle function in aged mice, as absolute and also specific force were increased, significantly increased, both and in vivo during grip strength test and also ex vivo in isolated intact EDLs that were studied in a setup in which muscles are mounted on a force transducer. Structural studies carried out in aged and age-trained mice are summarized in this slide. And what we found was that the perfect double cross-striation visible in adult fibers and lost in some fibers from aged mice was completely restored after long-term training. And our electron microscopy studies confirmed how in age-trained mice, exercise was able to improve mitochondrial disposition, but also exerted a positive effect on structure and positioning of triads. Our qualitative observations were confirmed by the quantitative data shown in these bar graphs, and these data were able to confirm that exercise counteracted effectively these changes induced by inactive aging. So, our data on human biopsies and mice gave evidence that muscle activity improves or maintains the correct association between calcium release units and mitochondria. but two questions remains to be fully elucidated. The reduced muscle activity would have the same effect also in adult muscle? And is the uncoupling between the two organelles a reversible process? To test this hypothesis, we used a model of transient denervation in adult rats. Using a combination of confocal and electron microscopy, we studied EDL muscle from adult rats denervated for 15 days, a stage in which muscle are really still denervated, and denervated and plus reinnervated for 45 days, a period of time that allows nerve to reinnervate muscle fibers. What we found was that the ordered disposition of mitochondria and calcium release units that is visible in electron microscopy and immunofluorescence pictures is lost in denervated samples, as you can see either in the two panels. But reinnervation was able to restore the normal appearance of skeletal muscle fibers. These qualitative observations were confirmed by our quantitative analysis, and importantly, the reduced association between calcium release units and mitochondria, which was caused by denervation, was completely rescued when enough time was given to nerve to reinnervate the muscle fibers. To conclude, in this cartoon, we model changes induced by inactivity to the architecture of the sarcotubular system and to mitochondria. Calcium release units here in yellow and white, and mitochondria here in green, reduce in frequency and lose proper orientation and also association. We believe that the reduced association of these intracellular organelles contributes to the reduced efficiency of aged muscle to produce energy, and hence would result in reduced endurance and force. But our studies demonstrated that these changes can be almost completely prevented by exercise during aging, and that this process is reversible at least after a short disuse. So my final observation is that the changes we have observed affecting the structure involved in calcium handling and aerobic ATP production are caused likely by inactivity modern age per se. So I would like to thank all the people that contributed to produce the data presented in this slide at our institution, at the University of Padua and Urbino, and all of you for your attention. Thank you.
Okay, thank you very much. Very interesting, excellent work. We can see that in not only nuclei are mobile in the muscle, but also mitochondria. We know mitochondria move. Questions?
Can I have just one question? Did you not try doing immobilization instead of denervation to look for reduced activity and mitochondrial localization?
No, yes, we tried with the denervation either in rats and also in mice. We didn't do the denervation experiments here in Chieti, but we made the electron microscopy analysis of these samples.
But you didn't try immobilization instead of?
I tried, but I didn't get the right protocol. So I think that I had some problems.
Other questions?
It's a good idea, Gillian. We're going to try to do also immobilization. We should do it, yes.
Yeah, because it was, if you're looking at the aging, it would be interesting to look at the reduced activity as well. And that will really button your hypothesis up there. It's very nice work. I love your microscopy.
Yeah, but if I can say something, in humans, we have a look at the effect of inactivity a lot with samples that Helmut provided to us. So we are pretty much sure about it.
Yeah.
I mean, I believe that there is the same effect. I mean, with mice, we have tried with mice. I remember Laura was trying to fix the mouse to don't walk with, but wasn't working really. So
Whereas you have the bed rest, which is really nice. Yeah.
yeah.
Maybe we should try in-limb suspension again. Yeah.
You have to try suspension. It is much easier in young rat.
Mm.
Or just look at the humans.
Actually, we do not use.
No,
With
it's a
mice,
class.
it's very difficult.
Yeah.
No, it
Mice
is a
very
classical model. 20 years ago, all people were working on suspended rat. It is completely well known what happens. You may start from literature and then decide which kind of suspension, but it is not very, very stressing for the animal. If you suspend by the tile suspension
Okay.
in a small cage, so that the no cage, you have to have a ton.
Gillian, do you have other models in mind?
No, I think it would be nicer to do it on the humans in the limb suspension in the humans, and then look at the recovery period. I mean, then you've got the human samples, and that's
Yeah.
And many people are doing the limb suspension or bed rest studies. So I think these will give you the ultimate answer.
Maybe sorry, Marco Narici has the material. You may ask him to,
Exactly.
because he
Thank
has
you.
a lot of suspended.
Not fixed for EM, but we should start talking.
Yeah, I guess I think that he doesn't
Human
have.
samples are trained afterwards.
Do the experiment in your laboratory. It is much safer.
Yeah, I have enough people.
Congratulations to Laura and
Thank you.
Feliciano and Simona and all the people there.
Thank you, Ugo. Thank you, Ugo.